So in this lecture, we're talking about cellular respiration. And if you're reading the Miller and Levine biology textbook, you can go and check out these chapters here in Miller, Levine. They've got some information there for you. All right, let's carry on. So there is a balance of energy between photosynthesis and cellular respiration. So plants like to do photosynthesis, and they do it in something called the chloroplast. And that process involves absorbing sunlight energy and changing it into sugar. But there's a group of organisms called heterotrophs that can't absorb sunlight, like humans and dogs and cats and mushrooms and other things that aren't plants. Those things need to get their energy from mitochondria, which do cellular respiration. Okay, so they need to use these sugars and create their ATP from that. Chloroplasts are able to use sunlight energy and make ATP during photosynthesis, but also the other main thing that comes out of that is sugar, glucose. And then the mitochondria will use this sugar inside of the mitochondria through a process called cellular respiration to produce ATP energy for the cells to use. Okay, now oddly enough, look at this. That process will release carbon dioxide, and that goes back into photosynthesis. So these two processes are tightly connected, and as we go through cellular respiration and photosynthesis, you should really think, what is the connection between cellular respiration and photosynthesis? Right? What's the connection between between cellular respiration and photosynthesis? Right? And remember that it's the autotrophs. Let's do that in green. Autotrophs are the things that can capture sunlight, and the heterotrophs don't capture sunlight, they eat their food, right? Autotrophs are able to capture sunlight energy and make their own sugar, make their own food as sugar. They do that by making lots of ATP, as we'll see. But then heterotrophs, they don't have chloroplasts, they only have mitochondria. And so heterotrophs use that sugar by eating other things, either plants or animals. They eat these things and get the sugars from the stuff that they eat, and those sugars go into cellular respiration in the mitochondria. Right, so again, photosynthesis, these are our autotrophs. Cellular respiration. Autotrophs also do cellular respiration, but heterotrophs only do cellular respiration. Okay, and then we're going to talk about something called fermentation, which you've maybe not talked about before. And this is kind of, this is specific to low oxygen conditions And we'll talk more about that later. There's two types. There's alcohol fermentation and lactic acid fermentation. And we'll talk about which ones do which. And so the last thing to just kind of talk about, we've already talked about this one here, that photosynthesis captures sunlight and stores it as chemical energy. Remember that chemical energy is ATP and glucose. And glucose is a sugar. And so because of this relationship, all the food that we see is thanks to, all the food energy that we see is thanks to plants, okay? Or is thanks to photosynthesis, right? So that's plants, and then plant-like protists, because they do cellular respiration, and photosynthetic bacteria. Now, they don't have a chloroplast, so they just do that in their cytoplasm. So we don't need to really think. So photosynthetic bacteria don't have chloroplasts, but are important to understand that they do contribute to the food web. All right. Okay, so we're talking about cellular respiration, though. We're not talking about photosynthesis. We are talking about cellular respiration, and cellular respiration happens in the mitochondria. It happens in the mitochondria. So here's the mitochondria. You need to be able to describe the parts of the mitochondria. You also need to know what happens in each of those parts. Do we do that next? Not really. So let's describe what happens in each of these parts here, okay? Well, actually, as we describe the process of cellular respiration, we'll describe where they happen, okay? So the parts that you need to know, the mitochondria has two membranes. It has an inner membrane and it has an outer membrane. So you can see here, here's the outer membrane. Let's highlight that. So here's the outer membrane right here. And then here's the inner membrane, you can see. The outer membrane is smoother than the folded inner membrane. Okay, there's two spaces that you need to know. There's the intermembrane space, that's the space between the membranes. So the intermembrane space is right here. You can see it goes all the way around, right? So here's the intermembrane space through here, here, around here. That's the intermembrane space. It also wraps around here, here, right? It wraps all the way around. And then you also need to know the mitochondrial matrix, or we can just call it the matrix. Whoa. And the matrix is right here. It's the space inside the inner membrane, right? So all of this here, this is the matrix of the mitochondria. Right? This is the matrix of the mitochondria. All right, mitochondria probably came from bacteria that were eaten by ancient eukaryotes, and they never got digested. They actually provided a benefit to those ancient eukaryotic cells by making their energy for them, kind of boosting their energy production. And so because these organelles came from bacteria, they actually have their own DNA. It's called mitochondrial DNA, and it's a circular loop, right? So these pieces of DNA are loops of DNA. They are not linear like eukaryotic DNA. They are circular DNA. This is circular DNA, just like prokaryotes. So that's why scientists think that mitochondria came from a prokaryote during evolution long, long ago. So here, these are the pieces of mitochondrial DNA. You can see them around. There's one there, there's one here, there's one here. And they also have ribosomes, and here are the ribosomes. And these ribosomes are actually smaller. These are smaller ribosomes than eukaryotes. Smaller than eukaryotic ribosomes. So they're the same size, same as bacterial ribosomes. Okay, so that's another piece of evidence for why scientists believe that mitochondria and chloroplasts, as we'll learn, came from ancient bacteria that were absorbed by an ancient eukaryote but never digested. It just provided a benefit. Okay, so those are the pieces that you need to know. Now, the other things that you should see here is something called the electron transport chain and ATP synthase. These are really important parts to cellular respiration, and they are stuck into that inner membrane. So these little black dots in here, those are the electron transport chains and the ATP synthase molecules that are stuck into that inner membrane. You'll also notice that that the inner membrane is folded. Okay, and that's called the cristae. So these folds, these individual folds, are called the cristae. And what this is the folded inner membrane. And the reason why they're folded is that it increases the surface area of the inner membrane, which means that you can increase the number of electron transport chain proteins and ATP synthase proteins. Okay, this will make a little bit more sense as we talk more about the process of cellular respiration, but all you need to know right now is that these cristae, they are folded parts of the inner membrane, and their job is to increase the surface area of the inner membrane and to increase the number, which in turn increases the number of electron transport chain and ATP synthase proteins. Okay, so we talked about endosymbiosis theory. And remember, endo refers to into. Symbiosis is a relationship between two organisms where there is a mutual benefit. Okay, it's a mutually beneficial relationship between two different organisms. So the relationship here is that there was an ancient eukaryotic cell, and it acted as something called the host, or the home environment, right? And that host provided a nutrient-rich environment that a bacteria could grow in. Now, the aerobic bacteria means that it's a bacteria that breathes oxygen, right? So aerobic means uses oxygen. And it provided an efficient source of ATP energy for that host cell, for the eukaryotic host cell. So it was a benefit to the bacterium, to that aerobic bacteria, to live inside the host because it was full of nutrients and there was nothing to kill it. And it was a benefit to the eukaryotic host, right? It was a benefit to the eukaryotic host cell because the aerobic bacteria provided an efficient source of ATP energy for that cell. So they lived together in this really nice harmony. Okay, so that is the story about how an ancient heterotrophic eukaryotic ancestor ate an aerobic bacteria but decided to keep it rather than digesting it. That's kind of my long way of saying it. All right. So, like I said, cellular respiration has there's a bunch of chemical reactions that happen inside the mitochondria. And so here's the overview, okay? Here's the big picture for cellular respiration. It has 3 parts. Okay, you need to know these 3 parts in pretty good detail here. You need to know the first part is called glycolysis. That happens in the cytoplasm. And you need to know that the second part is called the Krebs cycle, and that happens in the mitochondrial matrix. And then the last part is called oxidative oxidative phosphorylation. And that happens in the mitochondrial inner membrane. Okay, so if we go back to that picture here, the matrix, out here is the cytoplasm. Let's go back to here. So here's the cytoplasm out here, right? Oops. Undo. And out here is the cytoplasm. And out here is glycolysis. Okay, now in the matrix is where we have the Krebs cycle. Okay, Krebs cycle happens in the matrix. And then, like I said, in the inner membrane space, in the inner membrane is where we have the electron transport and ATP synthase, and this process together is called oxidative phosphorylation. Oxidative phosphorylation. Okay, so you need to know where those occur. And we'll talk more about those in detail in just a minute. Now, the second thing the second big idea here is that the energy in cellular respiration comes from glucose. Remember, glucose is a sugar, right? It's a sugar. It's a monosaccharide. And so here's glucose. This right here is glucose, C6H12O6. This is glucose. That's our sugar. When we combine it with oxygen, then we get carbon dioxide and water on the other side. Okay, so glucose and oxygen gives us carbon dioxide and water. And in that process, we're releasing the energy. Remember the sugar has energy stored inside of its chemical bonds. It's got stored energy. Okay, and the mitochondria is going to release that energy. That energy has to go somewhere, and it gets stored as ATP molecules. Okay, it also gets stored as some other molecules that we'll learn about. NADH and FADH2. So we're going to learn about those, but the energy from glucose gets stored as ATP, NADH, and FADH2, and then these things get used for various reasons, okay? The products are carbon dioxide and water. Now you have to remember the law of conservation of mass requires that we have the same amount of everything on either side, so all atoms need to be accounted for. So if we've got six carbons on this side, we need to have 6 carbons on this side, right? If we have 12 hydrogens on this side, then we need to have 12 on this side. 6 times 2 is, if there's 12 hydrogens here, then we need to have 12 hydrogens over here. 6 times 2 is 12, so there's 12 hydrogens there. And if we have 6 oxygens here and we've got 12 oxygens here, then that means that on the reactant side of things, we have 18 oxygens. We need to have 18 oxygens over here as well. So 6 times 2, that's 12, and then 6 times 1, that's another 6, so there's 18 on both sides. So it's all good. We need to have the same number of atoms on both sides. So the total reaction, the overall reaction, the formula is 1 glucose molecule and 6 oxygen atoms, or oxygen molecules, are rearranged or react to form 6 carbon dioxide molecules and 6 water molecules. That's the overall net reaction. And as a result, what happens is that 1 molecule of sugar can make 38 ATPs, which is a lot of energy. It's a really good result for breaking that sugar apart using oxygen. So this is just one last time to remember that you have to conserve mass on both sides of the equation. So you cannot destroy or just create matter. You have to have the same number of each element, the same number of atoms for each element on both sides, on the reactant side and the product side. So here we've got 1, 2, 3, 4, 5, 6 carbons. Over here we've got 1, 2, 3, 4, 5, 6 carbons. On the reactant side, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 hydrogens. And over here we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 hydrogens. On this side we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 oxygens. And over here we also have 18 oxygens, as we already said, right? There's 18 oxygens. They're just arranged differently. Okay, and that's the law of conservation of mass. Okay, so you need to remember the overall reaction is 1 glucose plus 6 oxygen will yield 6 carbon dioxide and 6 water molecules. Okay, now the overall reaction for this is an exothermic reaction. Okay, it's an exothermic reaction, which means that energy is released, which we've said down here. Energy is released from glucose, and we use that to capture it, or we use that to store as ATP. So that the released energy can be used to do work. We call this free energy. Right, this is called, right, this equals free energy, right, and that is used to make ATP, NADH, and FADH2, which we will become familiar with later. Okay, the reactants in this chemical reaction have more energy than the products. Right, this is showing the energy here, right? So on this axis over here, sorry, one second. Remember, on this axis over here, we've got energy. Okay, so this is showing energy up here. That's the energy stored in this is stored energy in the molecules. And this is along this way here, this is the reaction progress, right? Showing that we went from reactants to products. And also you can see this here is this energy, the amount of energy here is the activation energy. And since the products have less energy than the reactants, they must have released that energy as free energy.